Okay, can somebody confirm, please, if you can hear me?
We can hear.
Thank you. Okay, good. I think now audience can see you as well. You can walk around if you like. And okay, thank you very much. Thank you very much, everybody, to be here for this very interesting talk from Professor Shamith. I'm sorry if I got your last name wrong. Samarajiwa. Yeah. So he's an associate professor at Imperial College, London, for genomics and computational biology. And today he's going to talk about the application of machine and deep learning using epigenetic state alteration to facilitate cancer early detection, subtype classification, and metastasis. So over to you.
Thank you. Okay, thanks for inviting me. The talks in the morning by the students were really impressive. Hopefully, so my lab, can people hear me at the back? Okay, excellent. So my lab works on inflammation, cancer, and aging. And we do a little bit of metabolic diseases as well. And we are primarily a computational biology lab. And underlying, like all these diseases, our focus is to understand the rules governing gene and epigenome regulation. And so we work across different cellular processes. So we look at lots of different epigenomic dynamic processes. So all these things in the green. So the epigenome is important because that's where the environmental information gets transferred to the cell. Then we look at transcriptional regulation. So transcription, I know lots of you aren't biologists, and so basically, all of you have heard of DNA. DNA gets translated to transcribed to mRNA, and that process is tightly regulated. So these where genes get turned on and off. And then mRNA gets translated into proteins. So proteins are the things that do all the work in cells. And so we work in the translational space as well. So lots of work on translatomics, and we do a little bit of lipidomics as well. So understanding how lipids do various functions in the cell. This is the word cloud that basically shows all the different things the lab does. So we are a data-heavy lab. We basically focus on what's known as omics. So genomics, epigenomics, transcriptomics, proteomics, and metabolomics. So these fields, these areas generate huge amounts of data. Genomics alone these days generates zettabyte amounts of data each year. So it's a massively data-heavy field. And so we had to basically use data science and computational biology and bioinformatics methods to understand what these data sets mean. Most of my talk today will focus on epigenetics. So all of you know about genetics, right? All of you probably in school have heard of Mendel and his laws of how hereditary information gets passed between generations. Epigenetics, like I said before, is where the environment influences the cell. And it's defined as chemical changes, which are usually reversible, that influence gene activity without altering the DNA sequence itself. So you know about mutations that cause all kinds of diseases, and those things alter the gene sequence or the DNA sequence. But methylation are chemical modifications on DNA or other processes that doesn't alter the sequence, but alters the function of that fragment or those fragments of DNA. So environmental factors like diet, exercise, chemical exposure, stress, and the millions of these things which are called the exposome influence gene expression, like how genes get turned on and off through these epigenetic changes. So one type of epigenetic change is methylation of DNA. So methyl groups are tiny hydrocarbon molecules that get attached to base pairs in the DNA. So you know about the 4 different bases that make up DNA, right? Adenine, guanine, cytosine, and thymine. The cytosine bases can be modified by these methyl groups. And that's a way of marking certain bits of your DNA so that it basically changes the function of those regions. There are enzymes called DNA methyltransferases that add these methyl groups, so these CH3 groups, onto a particular position in the cytosine bases. And then there are other enzymes that can modify this into a hydroxymethyl cytosine group. And so marking your DNA at like these very, very well-defined positions changes the function of that region of DNA. And there are lots of microarray-based technologies, which are basically glass chips with bits of DNA probes stuck onto them, where you can then add your cell sample and it'll stick complementary fragments. Or you can use sequencing, where you can then detect where these methylation changes are. This type of epigenetic technologies have been around for a long time. People have been studying epigenetics for more than 50 years. Most of my talk is going to focus on cancer and how we are going to use these epigenetic changes or epigenomic changes to figure out what's going on in tumor cells. So cancer is one of the most challenging human diseases, or like animals and other creatures get it as well. And worldwide, there are about 19 million cases each year and about 10 million deaths annually. So it's not a trivial disease. The other thing is it's not a single disease, even though we give it a single name. It's a collection of 200 plus diseases. So that's one reason there's no cure for cancer, because you have to cure each single one. And out of these 200 different diseases, each of these cancer types have subtypes. So it's close to a collection of a few thousand diseases. And so because of all the work done in cancer research in the last couple of decades, most cancers can be treated effectively or even cured if you detect them really early. And so my first part of my talk is going to be on how to detect cancer early using deep learning methods. And I will tell you a bit more about what these, what kind of epigenetic changes we are looking at, what hypermethylation and hypomethylation mean, what CpG islands are in the next few slides. So, so methylation changes can turn gene activity on or off, right? And so that's why it's important to know where these changes are and what happens when you add a methylation mark or remove a methylation mark from a fragment of DNA. So most of you have heard of genes, right? There's 20,000 protein coding genes in the human genome and about another 30,000 non-coding genes. The human genome is about 3 billion bases long. So it's about only about 5% of the genome encodes for actual genes. The rest of it, people thought was junk DNA, but not anymore because we know there are lots of really important stretches of DNA that regulate how genes get turned on and off. So these stretches contain control elements. So at the start of genes, there's a control element called a promoter, and it's usually within the top end of the gene, the 5' end of gene. So these are like on-off switches, right? Like a light switch, you turn it on, light turns on, you turn a promoter on, a gene gets expressed, it makes mRNA. There are different types of these switches. And again, I'm going to only talk about one particular type, which is the CpG island promoter. CpG islands are basically stretches of C and G bases, can be about 300 to a few thousand letters long at the start of gene. And these things have like a co-island stretch, and you have like shows, and so that they have like particular pattern, basically. And these patterns can be detected computationally. So because they have these C bases, they can be modified using methyl groups. And so there are enzymes that can come and put on these methyl groups of the promoter and turn the gene off. Then there are other enzymes that come later on and take this off, and which basically makes the gene more ready to be turned on later on when other signals come in. The gene body itself can be methylated, and that basically increases the ability for the gene to be turned on. So it basically activates gene expression. Have I lost everyone so far? Okay. So because it's a student symposium, I picked two projects that students in my lab did. The first story was done by a PhD student, Issy Newsham, and a summer intern, Martin Sendera. And so Issy just finished a PhD, and she was my last Cambridge PhD student. Okay, so the aim of the project was, can we predict cancer or non-cancer from tissue biopsies? And we also wanted to predict the cancer type. And we want to look into the biology behind these model predictions, right? So our data sets were DNA methylation data from cancer patients. And we had control samples from non-cancerous tissue regions from the same patient. And so the task was to come up with like a proper prediction model that can answer some of these questions that we had. Like, can we predict whether the sample came from a cancer, like from a tumor or non-tumor biopsy, and what kind of cancer is it? So basically, we collect, we use data collected from patients where people took the DNA and sequenced it. It goes through this process called bisulfite conversion, which helps you identify which of the Cs had a methyl group attached, so was methylated. The data was from an older technology, which is the Illumina microarrays. People still use this to do certain types of DNA-based analysis, but these days it's mostly sequencing-based data, where you can directly sequence the 4 letters in your genome. Because it's the imaging-based technology, we have these signal intensity values, and we compute this thing called a beta value, which is a measurement of intensity in both the methylated and unmethylated regions of probes. So a probe is a short fragment of DNA that's complementary to a defined region in the genome. And so you can have like a few million probes that will detect very specific locations, namely the CPG islands that I spoke of in a few slides back. We transform these into M values because M values are more homoscedastic. And the approach that we used is this. We do a lot of pre-processing. So this is like the most important step. If you just take the data and try and like use it for training, there's a lot of inherent noise and biases in the data. Like some of the probes don't bind to where they're supposed to bind because they were designed incorrectly. About 20% of microarray probes do this. So you have to like filter them out. And so there are all these pre-processing steps which make the data suitable for modeling. So once you do that, so we had 2 approaches. We tried binary classification, and we had multi-class classification methods. We also tried very simple methods like logistic regression and SVMs, because like why use why use more complex methods like deep learning if a simple regression model can solve your problem? These things weren't bad, but across cancer types, they gave very variable results. So they were not consistent. And so, yeah, and then once we trained our model, we tested on a fraction of the data set that wasn't used for training. And we then, once we had a model with good performance, we tested it on 9 different external data sets. And then because our model had part of the model used XGBoost, which basically gave us the features that the model was trained on, or model used for generating the classification. We could then take the features and map it onto the genome and map it to genes that were near the CPG islands and interpret the results of the classification biologically. Yeah, there are lots of other things you do with the data, like imputing missing values and the M-value transformation I told you about. And so we had data from 13 cancer types, and each type had its own normal data set. And so we had a set of binary models, and we had a multi-class model where we combined all the normal data sets into a single one. And so it basically learned 14 different classes. The data came from the Cancer Genome Atlas. It's got data from 50,000 patients, and from almost 70 different cancer types. We only looked at 13 because those 13 had all the characteristics like enough normal samples and other characteristics that we were looking for. So each sample had about 300,000 features or probes. And basically, these are the cancer types. They're shorthand for the long names down here. As you notice, the normal samples are a lot lower than the tumor samples. So it's an unbalanced data set. And so we had to come up with ways to deal with that. So I'm showing you our best and the worst case confusion matrices for the binary XGBoost results here. So this is kidney cancer, and this is esophageal cancer. This thing didn't have enough normal samples, so it performed really badly when it tried to distinguish between normal and tumor. These are the performance metrics. So we looked at area under the ROC curve, precision recall, F1. We also did Matthews correlation coefficient because it's an unbalanced data set. And as you can see, majority of the tumors, the model could classify them correctly. There's a reason why some of the precision recall curve data is bad, and it's to do with the normal sample and lack of normal samples. And there were adenomas, which I'll go into later on. So these are the XGBoost results. And then we looked at the multi-class model for the same type of data. And as you can see, the classification is a lot better when you use the multi-class than the binary models. There were both the ROC and the PR curves, the metrics were almost perfect. So these plots start at 90% or 0.9. Then we took the features that the XGBoost model thought was used for classification, and we fed them into a deep neural network. So the deep feed-forward neural network, basically, we used it in a multi-class model, and it had better accuracy than just using the XGBoost. So this two-tiered way of getting the features out of the XGBoost model and then training a neural network on that improved our performance. So then the next step was to test these things using independent data sets. We had 9 different independent data sets. These things were a lot smaller than our training data. And some of them lacked normals as well. So these are more real world type data, not data collected from a massive consortium. The binary XGBoost model did sort of well. So again, I'm showing the best and the worst confusion matrices. So this is breast cancer. It got 1 wrong. This is prostate cancer. It got 12 wrong. And then when we looked into why it did that, so the data came with annotation, right? And out of 9 out of these 12 samples were adenomas. So adenomas, we didn't train our original models on adenomas. So these are benign tumors that can eventually turn cancerous later on. So because the model learned the methylation patterns that give rise to cancer or the patterns associated with a cancerous state, it found some of those patterns in the adenomas as well. And so that's the reason for the misclassification here. The multi-class XGBoost, again, didn't do that well. There were misclassifications, and some of this is due to things like head and neck cancer is usually a metastatic cancer type. And so the actual tumors can derive from lung and other things, other tissues. And so because they had the patterns, methylation patterns associated with the tissue of origin, they basically got classified under those tissues. The neural network did a bit better, but it still had issues with the head and neck cancers. But overall, the performance metrics were good, except for prostate cancer, which had issues with adenomas, and head and neck cancer, which the tissue of origin is different to the location it was derived from. This is just a PCA plot showing where all the probes fall into. The probes classified by the deep neural network. Because we had the probes, we could map them. So the features are the probes. The probes can be mapped to the genome, and then we can map it, figure out which gene that probe detects, or the CPG islands near the gene. And so you could do things like biological ontology enrichment and pathway analysis. And most of the features that the multi-class model came up with contributed to hallmarks of cancer and pathways associated with cancer promotion, basically. We looked at this a bit more closely. So each dot is a gene that a probe that was classified by the multi-class model. And then we basically figured out which pathways they were part of. So there's about 24 pathways that always come up in cancer. So biological pathways are basically a collection of genes that are sequentially involved, either signal or regulate or have catalytic activity in a sequence. So they're basically connected together in some way. Again, pathways are not technically real things in cells. In cells, you get information networks. Pathways are just fragments of these, like subnetworks. And so biologists have characterized, oncologists have characterized these really well. And one of the surprising things we found was almost every single known cancer pathway was part of the feature set that are multi-cancer probes that were involved in the classification they basically map to these pathways. Excuse me. So this is surprising because these pathways were identified because there's mutations in these genes or like genetic aberrations like deletions or duplications in these genes. And the fact that methylation, which can turn a gene on or off, does the same thing a mutation normally does, was, though not surprising, hasn't been observed at this scale before. We then took all our genes that were involved in the classification set and built a network model based on all the cancer pathway information available. And so this is what you get. So all the colored, whether it's blue or purple, it's one of the genes that was used in the classification. And the purple ones are genes that are either oncogenes or tumor suppressors. So genes that are directly involved in cancer. So eMethylNet, which is a model consisting of a DNN trained on features learned from multi-class XGBoost model, definitely demonstrates improved performance of binary and multi-class XGBoost. So the availability of these probes contributing to classification and mapping these to genes help us identify biological consequences of methylation across multiple cancers. And so these genes map to cancer pathways that are normally disrupted by mutations, suggesting that epigenetic silencing can have a similar effect. So these are the performance table. Again, I'm not going to dwell on them too much. So the second story I wanted to tell you is something that's ongoing. So the previous one was published last year. This one is ongoing, and this one was done by 3 master students in the lab. So it's to deal. So we wanted to figure out how cancer metastasis is regulated. So metastatic cancer is the thing that kills you. It's not the original tumor. So you have like an original tumor, and they disseminate and send cancer cells all over. And usually like millions of these cells are released, but only a few turn into metastatic tumors. And we want to see what regulates this process. So the mechanisms that drive metastatic cancer remains poorly understood. And people have been trying to use large-scale sequencing to figure out if mutations basically cause these metastatic tumors. And there's no evidence so far that it's a mutation-driven process. So then the next question is it epigenetic? And there's more evidence saying that there's epigenetic factors that help drive this metastatic process. So this, unlike the other one, is more biology heavy. I'll try and explain things, and it's shorter than the other one. The data we deal with is multi-omic data. These are huge data sets. Complex, they come from sequencing different layers from cells and tissues. And so RNA-seq basically measures gene expression. Histones are type of epigenetic mark. So it's different from DNA methylation. This is another epigenetic process. ATAC-seq looks at accessibility. So DNA needs to open up. So in a cell, you have about 2 meters of DNA, and it's compressed into 2-micron space, and it's wrapped up in these disc-like structures called nucleosomes. And so when you want to turn a gene on, for transcription factors to bind and polymerase to create your messenger RNA, it needs to open up from these nucleosomes. And so accessibility is a way of detecting that. Hi-C is an experimental technique which can look at interactions between different regions of your DNA. So I told you about enhancers, which are control switches that are away from the gene. So they're basically volume control. Promoters, which are next to the gene, are switches. They turn genes on and off. Enhancers are volume control. They can be tens of thousands of base pairs away. They loop back because DNA is a flexible, dynamic molecule. It's not static. It's always moving around. It's kind of like a machine. Bits of it are moving around, interacting with things. So these long-range switches can contact a promoter and increase the volume at which mRNA is produced. We had to process all these different types of data, and each type has a bias, like biases. And so you had to clean them up, normalize them, figure out what each one means, and then integrate them. So to integrate them, we used knowledge graph technologies. So we built a metastatic knowledge graph. Yeah, so sorry, these were the 4 different data types. Once you process the data, there's all kinds of bioinformatics analysis, and then you can get biological outputs from each one. And we used Neo4j to integrate, build a graph-based model and integrate these 4 different layers. We looked at 2 different types of metastatic cancer. One is renal cancer. So renal cancer is slow developing. It takes 30, 40 years to develop, and by the time you detect tumor, it's kind of too late to operate. And even when you surgically remove, so at the earlier stages, if you try to surgically remove these tiny tumors that you detect, most of them are benign, and the surgical procedure causes more harm than the actual disease. And so the metastatic tumors can spread to places like the bone, lungs, and brain. We also looked at pancreatic cancer. So pancreatic cancer is one of the worst types of cancers anyone could get. It has very low survival rate. And so we looked at all these 4 different layers from these 2 different cancer types, and we want to see if there were like similarities between the switches that control the genes. This table shows the different types of kidney cancer data we had. We had like 2 non-metastatic and 2 metastatic samples from each. So each one is paired from a patient. We looked at the gene expression, and we looked at normal kidney and normal lung data, and this is like a PCA plot. So they are nowhere close to in the dimensional space. We looked at other kinds of cancers as well. So our 2 samples were basically clustered with Wilms tumors. So these are like really aggressive renal tumors, childhood tumors. We did gene expression. So we had because we had paired parental and metastatic samples from each patient, you could do differential expression. And so that tells you which genes are turned on or off in the metastatic sample compared to the parental tumor. And so, yeah, there are a few thousand genes that were different. The next layer is the chromatin data. So, like I said, DNA is wrapped up in these nucleosome structures. They form these chromatin loops called topologically associated domains, and they form these compartments. And And within those compartments, they form these chromosomal territories. And so that's what you have in a single cell. All your chromosomes nicely packaged in a particular order, and where bits of it are interacting with different regions. High C basically helps us identify these interactions. So if you wanted to see these digital control switches talking to genes, we need to use methods like high C. When you look at the data, so these are the chromosomes and the color intensities interactions, you can figure out similarities and differences between different chromosomes. And you can figure out where chromosomes have broken and attached to different regions. So these are called translocations, very common in cancer. So these arcs show part of the chromosome 7 is attached to chromosome X, 21 to 12. And so you can figure out these massive damaged chromosomal regions. You can then compute the differential interactions between the metastatic and the parental. So depleted interactions are in blue, and the enriched interactions are in red. And so I'm showing you like a single chromosome here. So this one region of the chromosome interacting with the same. So if you number your chromosome from 0 to whatever number, use 0 to whatever number, each red dot is an interaction between two regions. So I told you about topologically associated domains. These are like regions that are close by that interact a lot more than regions far away. And these form these rosette-like structures. And I guess all of you know about Matryoshka dolls. So these are these Russian dolls that fit inside one or the other. So topologically associated domains are like that. You have these TADs, which are close by regions, and then you have bigger TADs that enclose the smaller TADs, and so on. And so using high C, you can figure out these interactions. The important thing is the TADs basically prevent certain regions from talking to certain other regions. But when in cancer, these mechanisms break down, and so you have control regions that are not supposed to talk to particular genes being able to talk to. So that's what this plot shows. So this is in a normal like parental tumor. You have like well-defined TADs. The same region in a metastatic tumor have a lot bigger TADs, like more open TADs that allow, so these are the genes within that region. And so this particular gene is a differentially expressed gene that's only expressed in the metastatic cancers. We identified 400 of these by using these kinds of methods. And large chunk of this 400 were things called transcription factors. So again, transcription factors are proteins that help turn genes on or off. So they can bind to the promoter, the control region next to the gene, and either turn that gene on or off. And so we visualized the ones we found in this experiment, and there are lots of them, a lot more than you'd expect just by chance. The next bit was to identify the distal regions that interacted with these promoters. And again, we use these histone modifications, which is like another epigenetic change, to find these regions. So there's 80 different chemical modifications that happen on these nucleosomes that wrap up DNA. Because it's like 3 billion, like your genome is 3 billion base pairs long, and each nucleosome has 4 histone tails or 4 pairs of histone tails coming out. And each of these tails, so histones are proteins, amino acid sequences, and there are particular locations that can undergo certain kinds of modifications. And so these 80 modifications, the combinatorial landscape is massive. Solving the histone code is a Nobel Prize. And there are lots of groups in the UK and the US trying to do this, because that will basically tell us how to control genomes at a scale never before possible. So enhancers, like I said, are distal regions that are volume control regions that interact with genes. And so we had a workflow that we took the data through to figure out which bits were active enhancers. Yeah, these are the histone tails of the nucleosomes I was talking about. And so we had like ChIP-seq data, again, a sequencing method that can identify these histone modified regions in the genome. And so these are the two pairs, the parent metastatic and the parental tumors. You can see the signatures, and each red bit is a location in the genome, are very different between the metastatic. So the metastatic, the more activity of this particular histone mark. We then came up with a data integration strategy, and which helped us map these distal regions onto the genes they control. And you can visualize this kind of information. So this is like a tiny region with two genes. The entire genome is massive, right? So manually, it's almost impossible to go through this. But computational methods can basically integrate all this information and basically identify precise regions with all the different patterns that we need to find. So these are basically histone modifications at the metastatic and the parental, and you can see the differences at some other locations. The interactions are the high C interactions where long distance looping happens across DNA chromatin. And these two were differentially genes that were only or differentially expressed in the metastatic tumors and not the parental. And so you can basically find the collection of all the genes that were differentially expressed that had differential histone marks and differential interactions across all the data sets. Once you have these, you can then use the genes to do figure out what these genes are involved in. And the signatures we got were those involved in invasion and metastasis, obviously. But that basically tells us that our data processing was correct because we were looking at differences between metastatic and parental tumors. But it did give us like particular processes that are unique to renal cancer metastasis. And then we could build the enhancer gene network. And so this is what it looks like. The triangles are enhancers. The circles are genes or the promoters next to the genes. When we looked at some of these, we built our own text mining NLP method and text mined the 36 million PubMed abstracts and ran this gene list through it. And majority of the genes we identified were metastatic genes, known in different contexts. Some of them hadn't been previously reported. And so we have a collection of metastatic genes in renal and pancreatic cancer. And we have similarities and differences of the control elements that regulate them. I'm going to skip the pancreatic bit. The transcription, we saw similar amounts of transcription factors in the pancreatic as the renal, but there were different set of transcription factors. So the proteins driving these metastatic changes are different. The genes involved are different, but the control elements are shared. We had all this information in the knowledge graph, so we could query it. And the next step is to, and we could integrate external data sets, other annotation into the knowledge graph as well. And basically, we identified a set of enhancers that were shared between metastatic kidney cancer and metastatic pancreatic cancer. And the next step is to take the graph embeddings from an knowledge graph and use it to ask all kinds of different questions or make all kinds of different predictions. So that's where the project is at. Yeah, so these enhancers may be potential targets for therapeutics. And because these are shared enhancers between different tumor types, you probably could develop a set of therapeutics that target different types of metastasis, basically. And that's my last slide. Thank you for listening.
Thank you very much. Do we have a question? People online can also ask questions, and in the room. Yeah, Alexandra, do you want to speak in the mic? Then people can listen to you. Okay. Online.
Thanks a lot. Thanks a lot for the talk. I mean, extremely interesting. I had a couple of questions. They are both related to the first part. So I saw in the second part that at a certain point you use protein-protein interaction information. So it's very beautiful, the methylation network that you reconstruct. Have you checked the information that you get on the basically gene network against protein-protein interaction information about cancer?
Yeah, so the protein-protein interactions, I guess I'm talking about the massive network we had at this one.
Yes.
That came from cancer data sets. So they were external annotation.
So you directly have also information about the basically expressed product, the proteins that are associated with it.
Yeah, so the genes came from the classification method, but to figure them out how they fitted into known biological networks, we used known pathways and protein interactions and literature evidence. So again, we text mined the 36 million PubMed abstracts to do this.
That's beautiful. I had a first question regarding the data processing, because my understanding is that obviously when you need to build the models, you're using methods that generally will require you to have the same features basically as an input. But you have those promoters that they are different across the different cancers. So how do you actually create an input data set that is actually consistent in terms of features? Because I assume that the promoter methylation will have slightly different lengths and size and positions.
So because the microarray platform we used is similar across the data sets, we came up with, once we did all the cleaning of the probe sets, we came up with a master probe set that we then said this was for all the different data sets. So some of the data had, some of the samples had missing values. And so we imputed, those ones. And that's how I think it was kept consistent.
Okay, anyone else? If there's, yeah.
Yeah. No, okay. Should I have to do? Thank you so much for the presentation. For the first part, I'm just asking if this is only a cancer data, right? It's biopsies totally. Do you think that if hematology can be in to foster, for example, the prediction, for example, now without saying, for example, data analysis, cancer can cause low B12, for example, and diabetic people have the same. So having said that, if blood analysis, for example, on all these kind of CBC and all these kind of blood analysis techniques can really help in fostering the prediction for cancer?
So the reason we pick methylation changes is because it's one of the first things, one of the earliest things that happen in a normal cell on its journey to a cancer cell. So it's like a really good early indicator of what's going on. diseases like diabetes, I think, has similar process. So it's a good marker of early diabetes as well. The only thing is there are better tests for diabetes like early on. So as a biomarker or like an early indicator, it probably isn't that good because there are better methods available as an indicator of diabetes progression. So I guess our aim was, so there are these things called liquid biopsies these days. You can take a blood sample and you have like fragments of cancer DNA in it, which is really difficult to extract, but people are developing better methods. And you can use a modified version of this method to basically say you have cancer DNA floating in your blood, and it's this type of cancer. And that would be less invasive, and it could be like a really good early, I mean, doing a blood test would be pretty simple, right? People could routinely have blood tests, and you could basically track cancer like really early, and then you can treat it. And yeah, so that was like the motivation for lots of this work.
Thank you.
Thank you. I've got a question. You've shown us the peak calling and differential expression, analysis. Obviously, you would have used different tools for that. Different tools provide different peak calling and different gene calling. Have you tried different tools, and has this affected downstream analysis? Or
Okay, so for gene expression, so there are like 2 or 3 really good methods like EDGA or DEC2, and they give very similar results. So gene expression, I don't think these days, there might be minor differences, and those differences won't affect the final choices, final results. For peak calling, there's hundreds of peak callers. So these are basically things that find these ChIP-seq signals. And so there are hundreds of algorithms. There's no good one. We use the ones, we tested lots. So my lab's done lots of ChIP-seq over the years. And we basically use the most commonly used ones, which may not necessarily be the best one, but they're basically validated across tens of thousands of data sets. So we know the problems they have. And the fact that there's no single best tool, because it's a really difficult computational problem to solve. And like I said, there's hundreds of people that have attempted. And so the solutions vary, like there are decent tools and good tools and really bad tools. And once you tried a few of these, you know which ones to use.
do you see for us as a computational scientist to have potential there to contribute?
I think maybe using some of the newer technologies to develop methods that can learn these patterns and come up with better, identify where these signals are a lot better than the earlier statistical based methods might be. But the fact that the space is saturated with hundreds of tools that do the same thing isn't, unless you can come up with a method that is really good and does better than anything else out there.
Right. And if there's no more question, I'll quickly follow up another one. We know the cancer is an inheritance disease, and for the breast cancer, like as a child born, we can do for the breast cancer, for example, BRCA1, BRCA2 mutation. Do we have something similar for the epigenetic? Because they are also inheritance diseases that come from epigenetic and modifications such as histone modification. So do we have some mechanism from there? Did you do any work there?
So not all cancers are inherited. Lots of them are due to environmental. So obesity, lack of exercise, bad nutrition, environmental toxins, like things you encounter day to day, all contribute to cancer. Only like a tiny fraction are inherited tumors, like inherited cancers. So epigenetic changes, you have like normal cellular processes, like DNA methylation, histone modifications. That's like a set of programs cells are supposed to do. If you divert from this, you get disease. And so there are good ways of figuring out this, like how good a cell or a tissue is. Like, is it like more normal or is it diverged so that it's in a disease state? Yeah, so
Do you think there's a possibility of in a future developing such method to cure before something get developed even in
yeah, definitely. Like if we can do it with a less invasive, because things I showed you today were tissue biopsies and they're a bit invasive. But if you can have a blood test that can detect methylation changes, so majority of the fragments of DNA in your blood are from normal tissues. And so the cancer methylated fragments are like finding needle in a haystack. And if you had better methods, people have used this to find metabolic diseases as well, not just cancer, from blood tests. And so I think that's kind of going to be the future if we can come up, develop newer computational methods. Yeah.
I think we might have a question online. There's a question online. Are there any plans to investigate if any of your differential expressed genes could be used as biomarker in non-invasive sampling method, for example, blood test to detect cancer or metastasis?
Yeah, I think there's a possibility. So we collected all the known metastatic genes from public databases and the literature. And because we are using like slightly different methods, in addition to those ones, we find newer ones. So those could be possible biomarkers. And the approach can be used for other diseases as well. You need lots of layers of data. Just having gene expression doesn't tell you everything. But if you have all these other different layers, integrating all of those can basically give you a lot of information.
Okay. There's another follow-up question. Have you looked at the role of estrogen and its inflated levels in obese individuals with respect to its role in epigenetic changes?
I haven't. I mean, there probably are people looking at things like that, but yeah.
No, that's fine. Thank you very much. I think we thank the professor for your talk.
Thank you.